Our next speaker is Viktorija Juric from Ireland. And the title is Targeting Glioma Stem Cells in Recurrent GBM Using Selective CDK Inhibitors. And please, Viktorija,
hi.
You should share your presentation with us.
I presume you can see my screen now.
Yeah, it's fine now.
Yeah, great. So yeah, hi, thanks for the introduction. As Janko said in introduction, my name is Viktorija. I'm based in Royal College of Surgeons in Ireland, here in Dublin, and I'm a PhD student. And today here, I'm gonna present you a part of my project, which is specifically focused on targeting glioma brain tumor in adults. Unfortunately, diagnosis and deaths from brain tumors are increasing over years, and it's estimated that it occurs in around 300 cases per year in Ireland, 2,200 cases in UK, and over 17,000 cases per year in US. It is slightly more common in men compared to women, and despite all the efforts to treat this disease with starting with surgery followed with chemo and radiotherapy, unfortunately only less than 20% patients diagnosed with brain tumors survive beyond 5 years, with the median survival for GBM patients being estimated to 14 to 15 months, which is extremely low. I hope this all showed that new treatments for GBM patients are urgently needed and new therapies have to be investigated. So in my studies, we are studying as well GBM on glioma stem cells, which occur commonly with cancer cell populations. Due to their resistance to radio and chemotherapy, the proportion of the cells which are so-called stem cells survive the treatment, initial treatment, which in turn leads to progression of disease in recurrence of GBM in patients, which is usually, unfortunately, then fatal for the patients. There's been a lot of research done in glioma stem cells, and there are different and opposing findings for which marker is the best, which marker is representative. There is no single marker which can be used to stratify and isolate glioma stem cells in GBM. In my research, we are using two markers, which are CD123 and CD44, which have been shown in literature that a higher expression of both of these markers correlates with patient survival, causing the lower survival rates in GBM patients, both newly diagnosed and recurrent. So specifically in my studies, we are using CDK inhibitors, which gain a lot of attention in the last few decades, not just in glioblastoma, but in many other cancers, especially since they've been clinically approved for the specific subtypes of breast cancer. They gained fast clinical approval in 2015. I have to say here, the CDKs used in this study are mainly CDKs, not mainly, they are CDKs 4 and 6. When CDK4 and 6 inhibitors were tested in glioblastoma, they were either not effective or too toxic for the patients, and all the clinical trials so far didn't show to be successful. Here in my research, we took another approach. So we are using CDK7 and 9 inhibitors and CDK2 inhibitors. So specifically, we are using CYC065, which is a new drug studied in the last few years, showed to be very effective in a lot of blood cancers and some solid tumors. And lately, it has entered the clinical trials as well, which is very good news. So CYC065 specifically targets CDK9 and 2. By targeting CDK9, it basically downregulates some short-living anti-apoptotic proteins such as MCL1, which induces apoptosis, or by targeting CDK2, it inhibits the proliferation of cancer stem cells. Additionally, we are using THZ1, which is a specific covalent CDK7 inhibitor, which acts through induction of apoptosis as well. So first, we wanted to study the expression of key CYC and THZ1 targets in GBM patient data set. So for that, we use TCGA data set. And as you can see here, all the three targets are highly overexpressed in GBM patients compared to the non-tumor tissue. Additionally, their indirect target, MCL1 anti-apoptotic protein, is also highly overexpressed. And that was regarded as the subtypes, the current subtypes of glioblastoma made by Verhaak and his group. More interestingly, TCGA data showed that both markers, stem cell markers CD123 and CD44 are as well overexpressed in glioblastoma compared to the non-tumor tissue. Additionally, they show to correlate with the patient survival, even though I have to say here in TCGA data set available to us, the number of recurrent patients is extreme. Well, it's very low, and that data set probably needs to be expanded to make bigger conclusions. So here we first then, in collaboration with our partners at Erasmus Medical Center in Rotterdam and ICM in Paris, we established a panel of 10 patient-derived cell lines coming directly from newly diagnosed or recurrent patients. They were established either in 2D model using ECM or 3D neurosphere, we call them gliomasphere cultures. So initially we studied the expression of these two markers in panel of our 10 cell lines. As you can see from here, these two markers are heterogeneous expressed in the panel of these cell lines, but we were specifically interested in recurrent glioblastoma. And as you can see here, they have a higher expression of CD133, which is considered to be maybe better predictive marker for GBM. So from here, we focused on recurrent glioblastoma cells and targeting CDKs in these cell lines. So initially, when we studied the initial response of these two cell lines to CYC and THZ1, as you can see from here, both of the drugs did have effect on cell viability when they were grown as glioma spheres, and this is 72 hours treatment. Even visible from here is that GTC9 cell line was more sensitive to the treatments compared to GTC10 cell line, which is our other patient. And on the other hand, THZ1 was less effective in, much less effective in GTC10 cell line. Now we went into the mechanism behind this and to see what is the possible reason for this differential response. So we first studied how do our drugs CYC065 and THZ1 affect the expression of stem cell markers. So first we showed that in both cell lines, CYC065 and THZ1 decreased the expression of CD133 marker. But interestingly, when we checked for CD44, we did not see significant changes in the marker expression. So we were thinking that might be due to the localization of the markers itself within the sphere. So our theory was that if CD44 positive cells are mainly inside, maybe due to drug is not able to come inside and actually target them. So what we did next, we used a really nice method called light sheet fluorescent microscopy, where we would embed our spheres in agarose and could stain them and image them in from different angles and get these nice 3D projections. But after studying this, as you can tell from here, our theory actually showed to be wrong, and due to the expression of I hope you can see the videos, but due to the expression of CD133 marker mainly inside the sphere in GTC9 cell lines, we could tell that our drug is actually targeting inner sphere areas. In GTC10 cell lines, it was hard to make any imaging conclusions on the imaging data because CD44 expression is much lower compared to CD133 expression. Then we next wanted to show the mechanism of potential cell death or reduced cell viability in these cell lines. And this is basically what CYC and THZ1 are supposed to do in this, well, not just in glioblastoma, but in other cancers. So basically they would reduce the phosphorylation of serine 2 and serine 5 residues on carbon.
Szczególnie, że to wiesz, to jak już skończę i to i tak będzie tutaj sekwencjonowane, no to...
Yeah, I'll continue. So they reduce the phosphorylation of carboxy terminal domain residues, which leads basically to downregulation of many proteins, but one of them we are interested in is MCL1, and that in turn triggers apoptosis, caspase activation, and finally apoptosis. So when we followed that in our 2 cell lines, we showed using an XNPI staining significant levels of apoptotic cell death upon 72-hour treatment with CYC and THZ1. Mechanism proposed on the previous slide was also confirmed on Western blot analysis for these specific targets. Additionally, we wanted to see whether this cell death induced in this cell line is caspase dependent. So we use a pan-caspase inhibitor called QVD, and we would pre-treat ourselves for 1 hour with QVD, and upon treatment with CYC and THZ1, we show that the cells are actually rescued from cell death, which would mean that the cell death induced in these cell lines is actually caspase dependent. That was also confirmed on Western blot analysis, where you can see in the presence of QVD, the cells are rescued from cell death, and caspase 3 executioner caspases are not activated. Similar results were found in GTC10 cell line, with the difference that THZ1 alone, 72 hours post-treatment, did not induce significant levels of apoptotic cell death. That was very interesting for us, and we went to study why. And as you can see here, CYC induced the same effect as in previous cell line, but THZ1 did not have effect on major players in this game. So we went to check for cyclin E. Due to the complex of cyclin E and CDK2, there additionally can target MCL1, which destabilizes MCL1. But due to the nature of THZ1 mechanism, THZ1 targets only CDK7 and does not target CDK2 because it's a CDK7 specific inhibitor. So we saw that in this time point, just targeting CDK7 in this specific cell line is not sufficient to induce significant levels of apoptotic cell death, and that's probably due to the compared to CYC due to their additional targeting of CDK2. Also, when we did the same as last time at 72-hour time point, we showed caspase-dependent apoptotic cell death upon a treatment with CYC065, but not THZ1. That was also confirmed on Western blot. But then next we thought, since I haven't presented all data here now as well for newly diagnosed cell lines, but when we studied the response to our treatment response and different characteristics of the cell line, such as protein expression, which actually did not correlate with the treatment response. But when we compared treatment response to specific treatment and doubling time of our cell lines, we found a very strong correlation between those two. So we decided to treat our cells, GTC, which is THZ1 resistant cell line, for a longer time, because that should then induce apoptotic cell death. And indeed, that's what really happened in our cells. And actually all the cell lines that were resistant 120 hours after treatment do respond to treatment with CYC065 and THZ1. That was confirmed on apoptotic death assay and Western blot analysis by cleavage of activation of executioner caspases. So we next went to see what is happening when our patients and patient-derived glioma spheres are treated with standard of care, which is at the moment temozolomide. Obviously, I don't have to say that combinational treatment for this kind of patients is urgently needed, since temozolomide alone is not even working in vitro, and it's not really helping patients that much, and nothing in clinic has changed since STUP protocol was established in 2005. As you can see also from here, TMZ alone did not induce any significant levels of apoptotic cell death in our glioma spheres enriched in stem cells. But on contrary, CYC065 and THZ1 alone induced really significant levels of apoptotic cell death. We didn't see significant additional or synergistic effect when these CDK inhibitors were combined with temozolomide. But yet we didn't want to give up on that since TMZ is used in clinics. So what we did next, we wanted to try if we have a range of increasing concentrations of both, can we at any stage with different concentrations reach synergy? But unfortunately, in these cell lines, I'm just going to show you an example for GTC9 cell line, we did not see any synergy or even additive effect of these two drugs, at least not in concentrations of temozolomide that would be clinically achievable. So next, we were interested to see whether these cell lines, which are sensitive, as we show now, to our drugs, are maybe dependent on any anti-apoptotic proteins from BCL2 family. I'm just going to quickly introduce you the method. So you probably all know, but BH3 profiling is a method used to kind of predict the dependencies on specific anti-apoptotic proteins. So it's a very nice method now established with basically a very nice machine with a plate reader where you prepare the plate in a way that you permeabilize your cells using digitonin, and then we next read them with pro-apoptotic peptides, which are derived from the sensitizers BH3 proteins. And basically we measure cytochrome C release, and that gives us this BH3 profile, which would then tell us whether your cell line is dependent on any of specific anti-apoptotic proteins or not. Interestingly, when we tested that in our cell lines, I'm just going to show you results for one cell line. As expected, we showed that our cell line is dependent on MCL1, which is showed with priming with noxapeptide, compared to DMSO control and our positive control, which is FCCP. We next wanted to exclude all the possible off-target effects, and we used MCL1 specific protein inhibitor to see whether we'll get the similar effect as with using CYC065 and THZ1. And really when we use S63845, which is specific MCL1 inhibitor, we got very similar results in terms of what we got with our CDK inhibitors. Furthermore, we used siRNA knockdowns of MCL1, and upon MCL1 knockdown, even in the terms of morphology, we could see the significant decrease of the sphere size and number of spheres in the cells in spheres with MCL1 knockdown. Additionally, that was also followed obviously with increase in apoptotic cell death, which was also confirmed with caspase-3 cleavage. We then were interested to study can our drugs be used in a more relevant setting. So for that, we first studied the colony formation assay and invasion properties and colony formation properties of these cell lines. And we showed here that our drugs stop the colony formation even when used in concentrations lower than IC50 for both drugs and both cell lines. Additionally, we were very happy to see that our drugs indeed prevent the invasion of the spheres into the matrigel due to the highly infiltrative nature of glioblastoma that makes it very hard for the surgical removal. So if we could find a drug that can stop the invasion of these cells into the normal brain tissue, that would really help in clinics, because then surgeons would have more chance to take as much tumor as possible, which could then reduce the chances of tumor recurrence. So we were very happy to find out that it indeed stops the invasion of the spheres. And this was only 24 hours post-treatment, so really promising results. So further on, we wanted to test how would these drug affect so-called normal brain cells. So for that, obviously we had to use mouse brain cultures, and we showed that both of our drugs, 72 hours post-treatment, did not significantly reduce the viability of mouse primary brain cultures and individually mouse cortical neurons. Additionally, we followed the morphology and the viability of mouse cortical neurons using calcine staining. And as you can see from comparison of DMSO control and our treatments, they really did not affect the viability of neurons. So this all showed us that they showed to be very, very promising results in vitro. And further on, we wanted to establish a model to study the efficacy of these drugs in a more relevant in vivo model. For that, we used chick chorial and towing membrane xenograft models, where you basically grow tumors topically on the cam of chick embryo. And I'm just going to show you shortly. So this is how the experiment is done. It's, I would say, not such a common method, but lately it's been used a lot. And I even saw that we have today 2 very interesting talks on this topic. So I'm really looking forward to that. So basically, after opening the window of the egg, and on the day 8, we would implantate the cells on the cam of the chick embryo and treat them and harvest them for further analysis. So I'm just going to show you results for one recurrent cell line. So here we show that upon excision of the tumors, that the CYC and THZ1 specifically, especially CYC065, reduced and inhibit tumor growth in vivo. So now it's a bit hard to make any conclusions from this because these tumors are inoculated with matrigel. So some of these parts that you can see, you might be thinking it's a tumor, but actually it's matrigel. And when I would touch them with forceps, you can see the difference between them. So tumors would be more solid, whilst matrigel would be very jelly-like. But because this is not really conclusive, we next have to do molecular analysis, and we did IHC analysis, where we showed that CYC and THZ1 treatment compared to control treated groups did actually inhibit the proliferation by following Ki-67 marker, and increase the apoptotic marker, which is cleaved caspase 3. We were very pleased with those results due to we were really able to grow tumors and then test the drugs in the tumors without not wasting, but without using a lot of animals and doing big animal studies. And this method is really great for big screens and kind of quicker screens of the potential drug candidates. So we went next into studying more stem cell properties in these cell lines. So what we did, we sorted our cells. And even here, if I show you this slide, you can see that we could observe differences between different sorted populations in morphology-wise in the sphere formation. Next, we wanted to see when we treat separated, isolated populations with our drugs and temozolomide, can we see any differences in terms of apoptotic cell death. So I'm just going to quickly show you some of the populations. So in CD133 population, we could see the increase in annexin PI and annexin and PI positive cells, and decrease in mitochondrial potential upon CYC and THZ1 treatment. Additionally, TMZ alone did not induce the same effect and was rather similar to DMSO control. On the other hand, when we combined them, we did not see significant difference compared to single treatments alone. So the similar results were found also in CD44 populations, even more obvious because they form bigger spheres, which you can see in DMSO and temozolomide controls. We were interested then to see, but this is just preliminary data, and we do have to expand on these experiments, but we were interested to see whether the cells which are isolated and then grown in culture change their properties. And indeed, we were very surprised, well, not surprised, but it was very interesting to see that the cells which were double negative upon isolation actually did become CD133 and CD44 expression. We can't really make bigger conclusions here because experiments need to be repeated, but we can see that they are changing their properties. The same happened with, this is a bit now, yeah, you can see here with CD44 positive and especially with CD133 positive, which gains some CD44 positive cells. So I'm just going to conclude, and just to summarize, we showed that CYC and THZ1 induced caspase-dependent cell vet in recurrent GBM, which are resistant to temozolomide. Additionally, we showed that those cell lines are MCL1 dependent, and more important, we showed that these CDK inhibitors reduce tumor growth in vivo model. Also, we showed that CYC065 and THZ1 induce significant levels of apoptotic cell death in CD133 and CD44 positive subpopulations. I would like to thank you all for your attention. I would first like to thank two patients and their families for who gave the samples for this research. Everybody from RCSI who were involved in this study, and our collaborators in Erasmus and ICM. Thank you very much, and I'm more than happy to take any questions.
Thank you very much, Viktorija. Well, we have time for maybe one or two questions, if there are questions. Well, is there any on the chat, or somebody wants to ask directly? Well, maybe I missed. These are the small molecular inhibitors, I suppose, not the antibodies.
No, yeah, they are small inhibitors, yeah.
Mm-hmm. And they are structurally much different or similar, just regarding these different effects. That's why I'm asking.
Different effects coming probably from the different CDKs they're targeting. So CYC065 is specific for CDK2 and CDK9, while CDK7, THZ1 specifically targets CDK7. But in the terms of mechanism, in general, they're both transcriptional inhibitors. So we do see differences, but that is probably also because of the different levels of CDKs, which are expressed in different cell lines. But they seem very promising. CYC065 is currently in clinical trials in a big range of cancers, and additionally they both cross blood-brain barrier, which would be very important for glioblastoma patients.
Okay, well, unfortunately we have to go on.